Isolated RBD is when we dream during the night, we're paralyzed. So some people lose this sleep paralysis that is normally occurring during REM sleep. And we know that those patients are at higher risk of developing Parkinson's disease or dementia with Lewy bodies. And by virtue of being neurodegenerative disorders, both dementia with Lewy bodies and Parkinson's disease do not appear overnight. So the brain changes that we see in Parkinson's disease or dementia with Lewy bodies already start to appear years before someone has a clinical diagnosis. So isolated RBD is a phenotype that we can study to understand how those brain changes appear in someone. And we published a few papers in the last years showing that patients with isolated RBD, so patients who haven't yet converted to Parkinson's disease or dementia with Lewy bodies, but who have sleep disorders, already show brain atrophy. So it manifests as cortical thinning, increased surface area, and other changes in the brain. We published a paper showing that these brain changes are associated with clinical variables, that we can use brain atrophy in isolated RBD to predict who's going to convert to dementia and who's going to convert to Parkinson's disease. And we also showed that we can recreate this atrophy computationally using a computational model of protein propagation in the brain. So we showed this for brain atrophy. So we showed that patients with isolated RBD have brain atrophy. So it can be recreated computationally based on the brain's connectivity and gene expression. However, in that paper that we published, we only used two genes, SNCA and GBA, because those genes are important for synucleinopathies like Parkinson's disease or dementia with Lewy bodies. However, it is very likely that a lot of genes are involved in brain atrophy in isolated RBD. So this study was about understanding the genetic underpinnings of brain atrophy in isolated RBD. So what we did is we took a large multicentric cohort of T1 weighted brain MRI scans from 171 patients with isolated RBD, which was confirmed through videopolysomnography and clinical evaluations, and 238 healthy controls. They all underwent MRI. We processed the scans to quantify atrophy in every individual, and then we performed imaging transcriptomics approaches to understand what were the gene expression components that predicted cortical thinning and cortical surface area changes in patients with isolated RBD. So what we found is, first, when we compared patients with isolated RBD to controls, patients with RBD had cortical thinning and they had increased cortical surface area, surprisingly. And then we performed partial least squares regressions to understand what were the genes predicting this pattern in the brain. And we found that the more a brain region was cortically thinned in RBD patients, the higher was the expression of genes involved in mitochondrial functioning, which we know is one of the reasons why people start having brain neurodegeneration and synucleinopathies. Regions that had increased surface area in isolated RBD had a higher expression of genes involved in the inflammatory response. So this is interesting because it tells us that there is a dichotomy in brain morphology changes in isolated RBD, thinning being related to mitochondrial functions, and increased surface area being related to inflammatory response. And what we did next, we wanted to know, are those changes mapping specific connectivity patterns in the brain? So we did structural and functional neighborhood analysis. So basically what we found is the more structural changes there is in one region in someone with RBD, the more there will be changes in a region that is connected to it, which support the idea that the changes that happen in the brain of someone with RBD are constrained by the connectome's architecture. So everything is connected and happens via the connectome. And we also demonstrated that it seems also to map on specific connectivity patterns. So for example, the atrophy changes were related to dopamine active transporter density, and it also mapped on somatomotor networks and default mode networks. So overall, what we show is there is already atrophy in patients with isolated RBD, and that these changes are constrained by specific gene expression and connectivity patterns. What we can say is the more a region in isolated RBD undergoes thinning, the more it expresses genes related to mitochondrial function. Now, it's hard to have a very clear idea of the mechanistic relationship between atrophy and mitochondrial dysfunction. However, we know from a lot of studies, even studies from the 70s, where mitochondrial abnormalities are already present in Parkinson's disease. Now, what is causing what? It's not very simple. Is mitochondrial functioning a starting point and then it creates atrophy, or some other mechanisms are causing mitochondrial dysfunction and then this leads to atrophy? We don't really know that for sure. However, what we can say is the regions where we see the atrophy in RBD are regions with a higher expression of mitochondrial functioning. And also, what is interesting is that in this project, we also used patients with Alzheimer's disease, just to be sure that what we were seeing was specific to synucleinopathies and not to neurodegenerative diseases overall. So when we take patients with Alzheimer's disease, we look at the atrophy, we do the same protocol, we see that the genes predicting the atrophy in Alzheimer's disease and the connectivity patterns in Alzheimer's disease are completely different from the one that we see in synucleinopathies and RBD, meaning that the association between cortical thinning and isolated RBD, synucleinopathies in general, so the relationship with mitochondrial dysfunctions are really specific to synucleinopathies and isolated RBD.